All right, ladies and gentlemen, last time in my house tonight, we're going to talk about HIV. We've already talked about viruses, and this set of notes is actually embedded in your virus notes. So you need to pull up your virus notes in another tab and then find the section that was on HIV. I skipped it in the last in the last screencast. I skipped HIV. Okay, so I'm in the section that says retroviruses. Retroviruses are a very specific kind of virus that has RNA and an enzyme called reverse transcriptase. And I told you that HIV is one example of a retrovirus. So now I'm going to tell you a little bit more about HIV now that we've done the immune system, and now we know a little bit more about helper T cells and the other cells of the immune system. All right, so the life cycle of HIV, how exactly does HIV work? First of all, number one, the virus docks, HIV docks at a CD4 receptor on a macrophage. What is a CD4 receptor, KJ? It doesn't matter. There's a kind of receptor on a macrophage that exactly matches the virus, the HIV virus, and it happens to have the dumb name CD4. That's what scientists called it. So if you look at the diagram, you can see that little orange spiky part on the virus kind of matches the yellow CD4 section. They fit together. It turns out that the macrophage also has a second co-receptor called CCR5. In the diagram, it's a green co-receptor. Those two together, the CD4 and the CCR5, are what allow the HIV to enter the cell. Now, this is an important aside. T cells also have CD4 receptors, but they don't have CCR5. So early on in the infection, early on in the HIV infection, the virus can get into macrophages, but doesn't affect the T cells, and that's important. All right, in step number 3, the HIV enzyme called reverse transcriptase. Now, it's in the macrophage. The reverse transcriptase reads the virus's RNA and it starts to make viral DNA. Reverse transcriptase, unfortunately, makes many mistakes. It's a super careless enzyme, and it makes all these different mutations. If you remember, when you're copying your DNA, if you make a mistake, you at least have proofreading enzymes that come back and correct most of the mutations, most of the mistakes. No such deal in HIV. Reverse transcriptase is a very careless enzyme and nobody proofreads it. So what that means is that the individual's own HIV is constantly mutating, constantly changing. Okay, in step number 3, the HIV viruses that are being made by the macrophage are released. And usually when we release viruses out of a cell, the cell blows up and the cell is destroyed. But interestingly and badly in this circumstance, I don't know if badly is a word. Anyway, when they're released by the macrophage, they're just pushed out through exocytosis and it doesn't harm the host cell. So what that means is that initially the macrophages are doing okay and the T cells are doing okay in the early stages of HIV infection. But the HIV is being replicated and replicated and replicated, but there aren't real effects on the immune system early on. However, as the infection continues, there are new mutations in the HIV because reverse transcriptase keeps making mutations. Later on in the infection, eventually some of the mutant HIV will now require a new co-receptor, and this co-receptor is called CXCR4. And unfortunately, CXCR4 is found on T cells. So now what it means is that these HIV viruses, which had initially been infecting the macrophages only and really not harming the macrophages, are now going to start affecting the T cells. And it gets worse from there. Unfortunately, when the HIV leaves the T cells, it leaves the T cells the way viruses usually do, which is by bursting out and causing the T cells to be destroyed. And remember that the helper T cells, which are a kind of T cell, are the generals of your immune system. And the helper T cells tell everybody else what to do. So as HIV starts attacking T cells, there start to be fewer and fewer generals, fewer and fewer helper T cells that tell everybody else what to do. So even if the patient has white blood cells, they sit around. They don't know what to do because nobody's there to boss them around. When it gets to that point, we now call that infection AIDS. So you can be HIV infected for many years, but once the T cell count really starts to drop and a person is no longer able to fight off infections, we call that HIV infection. We now call it AIDS, autoimmune disorder. Autoimmune. Oh my goodness, I suddenly forgot what AIDS stands for. Anyway, it suddenly becomes an AIDS case instead of just an HIV infection. So what it says here is the CD4 count. That means cells that have CD4 receptors. The number that you have or that a patient has falls below 200. A normal number within a certain volume would be 600 to 1500. So if it falls below 200, you can say that patient now has AIDS. What that leads to is certain opportunistic infections. Opportunistic infections are infections that we don't see normally in the healthy population. People with normal immune systems don't typically get these illnesses because their immune systems can fight them off. So with AIDS, people start to get infections that are less common. All right, that's it on HIV. Let me know if you have any questions. Have a great night.